the Australian institutes are using. So differential set is a set of lines which are having individual resistant genes in their blood. So only a single gene, so that exact single gene will give different phenotypic expression when you inoculate with different kinds of pathotypes. Individual pathotypes will give you different kinds of reaction, different scores. Based on that, you will identify what other gene is present. For example, I will give you an example for the gene postulation. This works on the principle of gene for gene hypothesis, what we know already. So you will get different kinds of reactions of differentials and the line of your question, the line which you want to know what gene is there inside it. So the next thing after the gene postulation, you know there is a resistant gene, you postulated it, you imagine, you guess that it is a new gene. What is your next step is to develop a population. You cross the gene, the plant which is having that particular gene with resistant susceptible plant, and you will develop mapping population. You can get, we have many number of options to develop mapping population. This will always depend on the trait of your interest and what is the genetics of that trait. It will differ when you concentrate on two genes, if two genes are there in the one line, or if it is a single gene is segregating, or if it is more than one gene, or it is having a QTL or like APR. You all know what is ASR and APR. APR is a major gene and QTL is the minor genes. So we have RIL, recombinant inbreed lines. From F2 you will go using single seed descent method. You will go from here until F6 and you will get the RILs. And BC1 F1 you go for one backcross and you just self it, the backcross, then you will get BC1 F2. We also call it as a BILS, backcross inbreed lines. And the other one is double haploid. It's quite easy when you have a protocol for transformation. You have a protocol for haploid culture and you do it as a colchicine doubling, and then you will develop a double haploid. In wheat, it is there, maize and wheat system we have for double haploid production that I will explain in future slides. So the choice of mapping population. Once we talk about mapping population, what we get? So why we can't go for mapping in parental population, or why we can't go for mapping in F2? F2 you can go for mapping, but until unless you have a replicate of it. So you can have a clone of the F2 and you replicate it and you can map it. But in F2 you can't go for replication, no, unless you do not have a clone. And the use of F2 derived F3 and BC1 F2, they have a different objectives of that. So you will be having one resistant parent and one susceptible parent. They are having a different kinds of blood in their this one, and you are interested in target gene, and you cross them. Blood will be same until this generation. If you pull P1 and P2 DNA and you pull RIL's DNA, it will be same. But there is a recombination is happening in the generations, generation over the generations. Genes, traits, and the markers, they are segregating within the through meiosis. So it will be recombined in the real population. Then you will look for what are all the markers which are co-segregating with your trait of interest. If you get one or two markers which are exactly typing your phenotype, then that would be a good validation for your work. Double haploid, as far as double haploid is concerned, there are many works have been done using double haploids, but only the lacking of double haploid is that number of recombinants you are allowing, recombination processes you are allowing it to until you develop it as a RIL. So there are 5-6 generations of recombination is happening. You are giving chance for the marker and the gene of your interest to go separated, but they are not separating and they are falling in the same blocks. So in double haploid, there is only one recombination which happens. So that's a lacking of that. But it's very quick process to develop the population. Once you have a population, then you will analyze. You have a mapping population F2 or F3, then you will analyze what is the genetics of that gene, whether it is a major gene, whether it is a QTL, whether one gene is there or two genes are there. Based on the segregation pattern, what you get in F2 or F2 derived F3 or BC1 F2. So I could not make this one on PowerPoint, so I wrote it. So if you see why we have to go for F3, F3 is very easy to when it is a single gene, and you need very less number of lines to predict. because you will be having one homozygous resistant if single gene is there, and a few F2 plants will be segregating 1:1, and you will be having a susceptible. It's a very easy one. So when you compare F3 and BC1, F2, there are two things. There are two genes in one resistant line. You develop a population F3 or BC2, BCF2, BC1, F2. This is for BC1, F2. So in the F2, what we get the gamete is 9 out of 16 will be resistant for heterozygote for both the genes. Both the genes resistance will be there. And the second one is resistant for the first gene, and the third one is resistant for the second gene, and the fourth one is it's susceptible. So these kind of segregation you will get, and you have to isolate single gene lines. You can't map two genes all together, and it will segregate in drills 1:1:1:1. So once you get into F2, you have to segregate. If two genes here, you have to isolate them into two single populations, single gene segregating populations. That will be very easy in F3, because 1 out of 4 will be resistant for first gene, and 1 out of 4 plants resistant for second gene. Here 3 out of 16. No? So number of lines which you need to extract the single gene lines is less in BC1 F2. And most of the cases, this is the donor parent and recipient susceptible plant. And when you go for BC1 F2, you will be having 75% blood of the recipient, the susceptible plant. So you are analyzing those two genes in the background of susceptible parent. So that will give you a different kind of information. And that can be taken forward if you want to make it as a population which can be for marker assisted. If susceptible parent is a good cultivar, very good variety as far as yield is concerned, and if you want to take it forward as a combination, then you can use this one. So double applied, I told, recombination is the issue in that. Once genetic analysis is done, you know whether it is a single gene or two genes. So you have a mapping population with you. So it's not moving. So next is you have to locate that gene. You know the resistant gene, you know it is single gene or two genes, and you have to locate the gene. Next is location of gene. Go back one step. Yeah, stop it. So you understood. In identification or discovery of gene, first is gene postulation. You know there is resistance, there is novel gene. Then you go for development mapping population for resistant line. Then you go So selective genotyping, nowadays everyone is doing, because 200 lines you have, the only thing is cost. We can't genotype. We can send it 200 lines for 90,000, 90K cheap, or you can't send it for 9K cheap because you do not have many. In India, we do not have 90K, we have 35K. 35K means we have 35,000K chip. So I have a mapping population, I have developed a reel, I want to send it, it is 200 mapping reel lines, and individually, so we have in India at the moment 35K it is coming to 13 lakhs per plate. Just you can genotype 390 lines, 80 plus lines in 13 lakh rupees. That's very cost, it's too cost for us. What we can do is you can select 10 of homozygous resistant, 10 of homozygous susceptible lines, and you genotype them for 90K or 9K. Then you will get the genomic that information. Then you put it together, and you can map it based on the polymorphism. So after knowing the location, like it is present on 7D, you know that it is the gene of your interest is present on 7D. So you have to map it. So in breeding, what happens is identification, mapping, and introgression. If you stop any place, at any place, it's useless. You will identify gene, you will not go for mapping, it's useless. If you identify gene, you map it, you don't put it into cultivars. You are not putting it, you are not introducing it into cultivars, and you are not taking it to the farmer's field, it's useless. So next step is the mapping. Mapping is something, you know it is present on 7D, but you don't know exactly where it is sitting on the 7D. It is long arm of the chromosome or the short arm of the chromosome. Then there are two methods of doing this one. If it is a major gene, it's very easy. You can do it bulk segregant analysis or selective genotyping. You will get the phenotypic data. You put genotypic data next to that. You will identify out of 90,000 markers, how many markers are sitting on 7D, because that location is on 7D. Then you will see how many number of SNPs which are sitting next to the gene of your interest. Then you will make a map of that. Then use those SNPs and you prepare your own primers for those markers, and you run it on the whole mapping populations. The second one is mapping QTL. Mapping major gene, you can do it under control conditions, under glasses conditions. Mapping QTL, you have to analyze it in multi-location trials or in number of years to get the accurate data. We have different kinds of QTLs, minor QTL if it is phenotypic variation showing is less than 10%, and major if it is more than 10%. And we have a lot of softwares. We normally use QTL cartographer, ICIM. It's very easy to use for anyone. Any one of us we can use if we have a genotypic and phenotypic data. So mapping QTL is very easy, even using bulk segregant analysis, if it is an additive kind of QTL. We have three kinds of QTL: additive, dominant, and epistatic. Additive is very easy to do, and followed by dominant, and next one is epistatic. So this is a kind of example I am giving you what we did there when I was there. So we screened and characterized 12 F3 populations. So what we did is we took germplasm. Earlier they had developed a population. They screened it. They did a postulation study, and after that they know that there is some resistant genes in these populations. I'm not giving you the name of populations because not yet published data, so we can't reveal here. So they were most of them were F3, and one was forwarded to F6. It was in F6. F3 you can predict the gene, how many number of genes. So for this one yellow rust, we predicted 2 genes may be there based on the segregation, how it is giving in F3. And one more population, we predicted recessive gene because it was segregating in 3:1. 1 is resistant and 3 is susceptible because it is a dominant gene is susceptible. So we predicted 1 gene here. So there were 2 lines I was using. They were the varieties of India, and they were segregating for some single genes. So we predicted single gene here. So the same population, repetitive of that population also gave us the second population of the same cross gave us its recessive gene. So these are the pathotype numbers, what we call then pathotype names, what we use in Australia, what we used to use. So this is the kind of recessive resistance what we got. That was the resistant gene. If you look at here, this is the kind of resistance, and the reaction was 1,2 double minus C. This is a resistance reaction. And here, next to that, we have susceptible plant. Because in F2, if you put seeds of a F2 plant, again it will segregate in F3 progeny. If it is single gene, it has to segregate in 3:1. In this case, one will be resistant, smaller, smaller, capital R, and capital R, smaller will be susceptible. So in the same pot, if you put 20 seeds, so you will get 4 resistant plants and 16 susceptible plants. That indicates that there is single recessive gene is present in that population. So we sent that for 90K SNP bead array, the F3 DNA. So we have got the information, and this gene is under naming process. We are naming this gene in future. So like that, I said 2 more lines which I was using. We expected some reaction, and we were expecting that YR4 or YR5 gene will be there. It was resistant. We don't know what is there, but based on the reaction, it may be YR4 or YR5, something like that. You will imagine it may be YR4, YR5. The best thing you can do is you run YR4, YR5 marker on that population. If it is giving negative for YR4 and YR5, that means this is something else than that. So you can develop mapping population and you do this one. So one more mapping like identification of marker. SR26 is a stem rust resistant gene which is effective against all the races of African UZ99. It's the aggressive pathotype available until now in India, TTKSK pathotype what we call. So we had 130 recombinant inbred lines of this one, and the resistant was giving 1 minus and susceptible was 3 plus. We wanted to identify a marker, SNP, codominant and repetitive across the laboratory marker for this gene. Then we sent that for TGBS. So in Australia, I said about 9K, I said about 90K. In India, we have 35K. They have 12K, one more marker array. That is targeted gene-based sequencing genotype array. That screens only for the genic regions, coding regions. So this map gave us 4 markers which were very close to SR26, and this SR26 came as a translocation from Agropyron elongatum. So this translocation was the large translocation in our set S, the plant which we used as a donor, and there was one more small translocation in wild abortive 1, so that CASP 224 and 225. These 2 markers are SNP based markers. You can run it in any laboratory for that matter is concerned. So we developed a marker. So what is next step? You have to validate it. So this marker we validated across the mapping populations which are having SR26 in there, which is segregating. So SR26 is segregating. Along with that, my project was having that to integrate this SR26 into Indian wheat cultivars. 3 cultivars I have taken. So once I did F2, once I went until F2, I have to go for back crosses. So as to select SR26 homozygous resistant plant out of segregating, we use this marker. Wherever you see this kind of color, orange color, it shows that SR26 is in homozygous condition, that plant. It is very difficult to differentiate this one and this one phenotypically. So this marker completely validated my population by showing the difference between homozygous resistant and heterozygote resistant plants. Next one. Okay. So if you see here, this was the parent 1, this was the parent 2. SR26 is coming from both the parents. Imagine if SR26 is coming from one parent, it will segregate and it will not be present in a homozygous resistant condition. If it is coming from both the parents, you will have a copy from both the parents. In the segregating, you will have, in F2 you will get homozygous resistant, homozygous heterozygote resistant and susceptible. So that is differentiated here. Wherever SR26 is coming from both the parents, you get this one, AA. Okay? And wherever it is not coming from either of the one parent, you will not get homozygous resistant. That means that this marker is exactly going with the trait of your interest. So we checked our material for resistance. We did a genetic analysis. We did population development for that. We did location of the gene. We did mapping of that gene. We identified the marker for that respective gene. So what is the next process is, use that gene and the marker. You have to introgress that gene by using that marker into your cultivars. So this was the project I had with Australia. So these were the steps for effective introgression, because I had it for 2 years. So within 2 years, we were having a lot of objectives to achieve. So we went for use of speed breeding approach and double haploid production to get the lines fixed for multiple genes. So we first tested effectiveness of target genes. Why effectiveness target genes is, material is for India. We have to use that resistant gene in India. You don't know whether that resistant gene is resistant to Indian pathotypes. Australian pathotypes is something different. Indian pathotypes is something different. Maybe that gene is resistant to Indian pathotypes also. So you have to characterize that gene for Indian pathotypes. We did characterizing that target gene. Then we developed backrust derivatives and double haploids. Then high throughput phenotyping we did for them, followed by screening of background genes. You have some foreground genes, but still your varieties will be having some other background genes that I will explain. Then you can develop a triple rust resistant. Can you imagine if you release a variety which is having only susceptible resistance to yellow rust, is it useful? Because there are so many areas like Delhi, we will get leaf stripe rust majorly and leaf rust in later stages. So you have to have multiple rust resistance, that we call as a triple rust resistant lines. So when you release those varieties, you will be having a surety that it will go very far ahead in breeding, production programs. So next you have to go for large scale multi-location testing. Even if you are putting a gene into your material, whether if it is not performing very well in the yield trials, it will not be useful for you. So the project was development of high quality rust resistant lines. We had 6 genes, YR51, 57, YR47 linked to LR52, SR26, 22 and SR50 for stem rust resistance. All these genes are effective against Indian pathotypes. We wanted to transfer these genes into these 3 Indian varieties, which were famous varieties, and few of them still in the breeder seed production pipelines. So this is the process what we followed. If you see in next slides, this is the overall plan of our thing. So Indian wheat varieties were these 3, and Australian donors were for resistant genes. And first we went for developing backrust BC2F1 and BC2F2. Then we screened them phenotypically, and then we did background selection for other genes. Then we ended up with developing double haploids, F2, and triple and double crosses, which are having triple rust resistant flavor in their blood. So we shipped that material to India. Last year we got it and we evaluated those materials. So before that, high throughput phenotyping is very important. High throughput phenotyping, because you have to evaluate large number of lines, and even you have to evaluate it against different countries' pathotypes. If you have to use it for other countries too. So individually these will differ as far as rusts are concerned. Stripe rust needs different temperature regime, and leaf rust needs separate, and stem rust also needs separate temperature regime. So they have very high throughput, not like in India we are still a bit far behind. So we can sow these in pots, peat pots, then you grow them in germination chambers. They have different chambers for germination. After 10-12 days of germination, 2 to 3 leaf stage, you will inoculate it. Inoculation also they have different chambers, mist chambers and all. Incubation you can do. Incubation chambers are different for stem rust, leaf rust and stripe rust based on the temperature requirement of that. Then you can shift to microclimatic rooms. They will be there until the development of the disease, and you screen them. So these were the markers. Once we did phenotyping of those, we did 2 levels of genotyping. One was genotyping for the genes of our interest, and second genotyping for genes of not our interest, but still they are segregating in our populations. We were expecting LR26 is there, SR2, LR34 it's adult plant resistant genes which is there, and which is the prominent genes used by most of the breeders in India. YR4 already there in India, this one, and YR15 it is efficient in Indian for stripe rust. So these were the markers which we used. If you are targeting any of these genes, you can use these high throughput markers. So SNP, I was talking about SR26, the marker. Earlier what we used to use is SR26 number 43. But in between of my program, we developed a marker, SNP marker, CASP224, that we used in our further studies. So you're doing introgression, glasshouse phenotyping and genotyping, but still you have to evaluate your lines for field conditions also, because what you evaluate inside the glasshouse condition, what you get outside in natural condition is something different. because there are a number of new pathotypes are keep on developing. We know in India for yellow rust, there are some pathotypes which are freely available. Some pathotypes already have been collected and they are contained not to allow them to be used by breeders to evaluate, and they spread outside outside in natural environments. So to evaluate our lines for natural available pathotypes, we did it. There were two locations. One was I was placed at Sydney location. So we took one evaluation program in Sydney, near Sydney. One was 520 kilometers away from Sydney. There was a place called Narrabri. We have a Watson Plant Breeding Institute there. And most of the Australian program, grain research program is happening there. They evaluate material there. We get a lot of rust there. And for yield also, you can evaluate your material. And you all know here we get labors and all to take care of our work. But there, you have to work all alone. Starting from sowing, observations, harvesting, you can sit in the harvester and you can harvest, bring them to the storage and you will store it. That's the kind of system they have, and it is very highly sophisticated system. So once you make the background selection of these genes, you will prepare the panel like this. So you know the first backrust line which you prepared for, developed for YR47, and you know in the background we have 3-4 more genes. SR2 is there, LR34 is there, and YR4 is there. This kind of genes you will use it for large scale crossing program. Then is hybridization. Once you have a single gene lines, then you go for hybridization. So if you see here, it was a single gene like SR22, 1 BC2 F2. We crossed with YR57. So it is having 2 genes in the F1 blood. Then F1 we grew it in the field in the main season, and we crossed to develop a mega F1. It is having SR2 genes, and this F1 is having 2 genes. You are bringing it together in F1. It will again segregate. What you do is you harvest the cross, mega F1 cross, and you sow it in the glass houses. We did a sowing in the glass house and under controlled condition in the off season. Individual plant is individual mega F1 seed. Individual seed will go into segregate, and you have to catch hold of that F1 where your all genes are segregating. That's the thing. It's very difficult to do a step every generation, but in one generation if you catch hold of, and we selected few lines which are having triple rust resistant genes, and those were genotyped, and they were selected and they were shipped to India. So this is the kind of lines you will develop. In the background of PBW550, there was a line initially SR22. We crossed it with YR57. F1 was having these 2, and there was one more YR47 linked to LR52. Leaf rust resistance is also coming together. We crossed these 2 and we developed a triple cross. And that triple cross, we grew it, hundreds of triple that F1s, and we selected for SR2247, YR57. The plant which are having all these 3 genes were selected. And that will be grown in progenies, and just next time you multiply seed and you will keep on processing that generation by generation in the plot. So you can take out best line out of it. So one more method was double haploid, what we call in wheat. We have a wheat maize system of double haploid. So there are 2 genes, one is in P1, one is in P2. You have to cross and you have to make backrusses, 6 generation of backrusses. Then you will get fixed line for those 2 genes. But in double haploid, what happens is you will cross wheat, which is having wheat F1, which is having 2 genes. For example, SR2226, just cross it. You will have a F1, which is having SR22 and SR26 together. You will take the pollen of maize, you put it on the wheat plant, wheat ear, stigma. What happens is, in the next generation, when the zygote is formed, the chromosomes of maize will be degenerated, okay? That is digested. So you will be having a haploid number of chromosomes from only wheat. So this embryo you get, that we call it as a caryopsis. And that caryopsis will be doubled using colchicine, then double haploid seed will be produced. It takes only 1 year. You imagine taking 3-4 years to develop backcross, and this can be done in 1 year if you have a sophisticated facility. For this we use speed breeding approach. What happens in speed breeding approach is you grow maize under controlled conditions. Maize cannot be grown in cooler temperature, you know that. So now if you sow it in November, it won't grow also. And wheat will be grown in cooler temperatures now in Rabi conditions. So if you want to make maize and wheat, it is very difficult in India. So you have to grow maize in controlled conditions, grow your wheat in fields, just you make the crosses and develop double haploids. This system is not yet explored much in, there is one institute which is doing, but it's not much explored in Indian conditions. So the next one is vernalization room we use, but you know maize flowering and wheat flowering is entirely different conditions. We used vernalization and light to control the flowering regime of wheat. Then you can cross maize and wheat and to develop a haploid where chromosome of maize is degenerated. Then you can culture that Caryopsis embryo in the tissue culture, and you can harden those plants after getting 2 leaves. Then you can treat with colchicine to multiply the chromosomes of wheat. Then you will have a double haploid, where these will be having 2 genes fixed in 1 year. So the overall program was this. We started with backcross lines. So high throughput phenotyping we did, then genotyping for the foreground selections, and background genes, other genes, APRs. And some quality work also we did. Then we developed through single crosses and double crosses. We had double haploids, which are already fixed, double crosses, triple crosses, still they are segregating and we are selecting in the field. And we have F2. All these we shifted to India and we are evaluating this. So we had taken multi-location trials at 2 locations, Sydney and Narabri. So if you look at here, the lines which we developed and shipped to India, this is the agrolocal susceptible. It's not very clear, but this one, see if it is completely clean from the disease. Nothing, not a single spot you can see on the this one. So this is also one more picture of that. We were growing them in the progeny rows. Now we have shifted them to plots, individual plots, larger plots, to analyze their yields. This is the kind of segregation you get in triple cross or double crosses, because too many number of genes are segregating. And phenotypically you have to select, genotypically it is very difficult to go. Disease resistance is something fantastic that it's phenotypically easily selectable trait. So this is a susceptible plant came out of this line, and this is a resistant plant which is very good with the performance for the plant type, and this can be discarded and this can be forwarded to the next generation. These are few other. So what is next? We have already shipped these lines into India, but there were some issues that few of the lines we could not ship to Australia due to some issues of plant registration. So we are taking up a project to transfer these genes again into leading wheat cultivars of 4 zones, because we have 3 kinds of rust resistant genes we are shipping them. Also double haploid we are exploiting in India. As I said, we are growing maize in phytotron. We are trying whether we can be successful in developing Caryopsis. At least we develop Caryopsis, then that can be pipelined through tissue culture protocol. So in conclusion, what I say is understanding all these concepts of wheat and the rust is very important. The kind of variation is present in the pathotype, how wider its adaptation is, and how aggressive the pathogen is going, like stem rust UZ99 for example, and deployment of major and minor genes together. If you are targeting only one gene for a particular rust, it will be knocked out one or other day in the future. There are so many examples happening even in India. So always it is better to go for major and minor the QTL's integration into one. So definitely high throughput phenotyping and genotyping are very essential, and they will take us forward in developing and delivering triple rust resistant wheat varieties in coming years. For references, if you wish to, if you want to have a basic information of wheat rust, so a paper came in 1995, wheat rust, an atlas of resistant genes by Bob McIntosh. Still he is 90, now he is in 92. He comes to office at 6 o'clock in the morning. He leaves at 11 o'clock. He is the one who names all the genes of different kinds of rust all over the globally. So there is one review from my supervisor there, and we have few publications, so that can be referred. Thank you so much. If you have any questions, you can. Finished in time? Yes.
Sir, why we select maize pollens for in double haploid? Only maize pollen.
So we have different system of double haploid production. Like we are trying for different crops, but as far as wheat is concerned, there are two methods until now we have exploited. One is crossing wheat with maize. What happens, as I showed in the slide, so you have a wheat chromosome. There are 42 chromosomes are there, 21 homologs. So when you cross two parents, wheat and wheat, okay, 21 chromosomes are contributed by your parent 1, 21 chromosomes will be contributed by parent 2. So they will come together, it becomes a exoploid wheat, 42 chromosomes totally it will be. So what happens is, when you cross wheat into wheat, you will get a complete set of chromosomes. So you want to transfer, there are two genes, you want to fix it. So parent 1 is donating one gene, first gene, parent 2 is donating second gene. So you will make crosses. So they will come in heterozygote condition. Then you will do back crosses to fix those genes, both the genes altogether into one line. So it takes 6 years of your effort, or 6 generations you need. Even if you use off season, you need 3 years. What happens in maize is, you have a parent, you just develop F1. No need to go for 6 back crosses. You have a F1, there is 2 genes, first and second gene, they are in heterozygote condition like this. So when you cross it with maize, okay, cross it with maize, maize will also contribute 2 genes, like its chromosomes. But that chromosome will be degenerated in the first F1 itself. Embryo will not be developed. That embryo will be a watery embryo, what you get, that we call it as a caryopsis. There you will not be having chromosomes of this one, yes, chromosomes of the maize. So it will be degenerated. You will have a haploid number of chromosomes of the wheat only. So in that condition, you have a F1, like of wheat and maize, you have a F1 where your genes are sitting in opposite positions, like both are in heterozygote. Then what you do is you treat it as a colchicine, because that is a haploid. You will be have a one copy of that chromosome. You will treat it with a colchicine. It will multiply, like it will duplicate. So one copy of this gene, one copy of this gene sitting on the chromosomes, all 21 will going to copy themselves. So that becomes homologous, like second copy is created. So those 2 genes will be fixed in your double haploid line. So maize is used just to create a haploid embryo of wheat. In a haploid embryo, both the genes will be there. They will be in heterozygote condition, but only one copy will be there. The second copy will not be there of homologous chromosomes. Then you double it with the colchicine. The second copy of all the chromosomes will be produced. Now that time, that gene copy also, it becomes a second time. So 2 copies of that gene you will be having, and that is fixed. You go on selfing the double haploid, you won't get changes in those genes because they are already fixed. They will segregate. They won't segregate in the population. Yes. Got it?
Thank you, sir, for wonderful presentation.
Thank you.
Really, you have done good job in Australia. I have a few queries, sir, here. Like you did introgression of different Western genes for us to Indian genetic background of the varieties. So what was the haploidy level of that Australian donor? It was hexaploid or tetraploid one?
No, all were exoploid land races.
And
Yes.
their sources were present in the single genetic background, or they were in different, you took in the single background?
First, actually, this work initiated through a project, ICAR-IARI project, what they had with Australia and India. There was a collaboration project. So their objective was to develop rust-resistant lines for India. using novel resistant genes which were identified in Australia. YR 51, 57, which I took, they were identified nearly during 2013-14. I went there in 2016. There was a student, Randhava. So now he is in CIMIT. So he identified those two in Australian land races. So those land races were crossed to Indian varieties. So first they developed BC2F2. When I went there, I got BC2F2 for my project. But by BC2F2, almost our lines were fixed. No? Like Indian background has been recovered there. So those lines I used. Those lines will be already performance-wise, yield-wise, they will be very good. No need to select for yield. But you have to select it for only foreground. That's why we didn't go for background selection, what we call background selection here. So you can avoid that one. If you do not have money, what is the other way to do? Run a project. What you will do is, you have a land race. People say foreground, background, foreground, background. But one more thing is you can do simultaneous backcross when you concentrate on two genes. Just you cross it with the land race, you keep on going for backcrosses. At the end of BC2F2, you cross those two. Already in both the single backcrosses, you have recovered the genome of your cultivars. So if you cross those two, they will be very good with the performance, yield-wise, agronomic traits, and they will be having those genes. Yes.
And how about that recovery in BC2F2, like percentage recovery from the?
Yeah, it was nearly 82%. That paper has been already published because that work I didn't do. Until BC2F2, they have done it. They have done the background selection for these lines and the foreground selection, and it's published in Agronomy Journal. So it's nearly more than 83% recovery was there. So 83% recovery, more than 83% recovery in all of these, and we crossed those backcross lines having different resistant genes in their blood, and we combined them to make them into triple rust resistant. Yes. Any more? I would have like presented that data, but it's already published on it. Someone said, so I could not take it.
There was a statement, crossing test cultivars with monosomic series. So how you make those how you take that?
We have already got monosomics. Earlier they used to use, now also people are using monosomics are available already. So maybe you have heard of Sears, they have developed in USA. So they have monosomic series for all the chromosomes in wheat. So you use those monosomic series, and when that chromosome is just, it's a principle of that chromosome is present or it is absent. You will have a monosomic line you crossed with your line of interest, that is particular chromosome is missing, and how much of resistance you are getting in the progenies. So if that particular chromosome is missing and you are not getting susceptible, you are not getting resistant. If it is present, especially that, for example, 5D, when 5D is there, you are getting more number of resistant plants. If 5D is absent, then you are not getting. That indicates that particular resistant gene is present on that chromosome itself. That's the principle of that.
Sir, is there somaclonal variation observed during the embryo culture, and what is the survival rate of this embryo?
Yeah, actually the main problem of double haploid production in wheat is survivability. And until making crosses is not an issue. You take a wheat F1, heterozygote it will be, and you cross it with a maize. Once you put it, that embryo, what we call caryopsis, there is a watery bulb, what we call tube, in that single 1 or 2 embryos you get. Once you culture it on a culture media, so few of them will die due to infection or something under the tissue culture laboratories, and few of them will generate, regenerate into 2-leaf stage. So once they are in 2-leaf stage, you can't grow it continuously on a media. You have to shift them to the soil and you have to harden them. So once you shift them to the soil, definitely many of them will die. So that recovery percent is too low. If you make 100 Caryopsis line, in Indian studies they get 20, 25. They will be survived as a haploid plants. Okay, so a haploid plant has to converted into a well-established haploid plant, and that should be treated with a colchicine. After that, again you have to cut the leaves, and again it has to establish and it has to produce seeds, haploid seeds. That's too much of energy. Even a plant will not be having that much of energy, and still you are giving that much of protocols. Definitely you will lose. But in Australia, they have a very good protocol, step-by-step protocol, and it is 24/7 work. They work until 9:30 in the night, because they have a indents from private companies to produce double haploids. They are not producing double haploids only for making these genes to fix in their populations. Double haploid for breeding purposes also. Like in normal breeding, what we do, we cross two plants, two better yielding plants. You will have a F1, and then you will get F2. What we do is, you will select individual F2 plants based on the performance of those plants. Then you will try to harvest individual plants, and you take until F6, okay, individual plots, that is 6 generations of selfing, to make that line fixed as far as F2 is concerned. F2 performance, if you see, that will be shown in F6. So instead of going for so many generations of selfing, F6 advancing this one and fixing that F2 plant, they use double haploid. Once they get the F1, they harvest the F2 seeds. F2 seeds will be converted into double haploids. What happens is, within 1 season, you will make that plant as same as looking in F6. That's the advantage of this. And yes, this is very problematic issue in that one, the regeneration. But still, they could get more than 50% of plants which can survive after tissue culture and after colchicine treatment and all. And ultimately, if you make if you culture 100 embryos of haploid, ultimately you will get 50, 55 embryos, double haploid plants. Yeah. Individual double haploid plant will produce nearly 10 to 20 seeds. So those individual seeds, you can put it in the progenies, and you can evaluate.
Sir, after colchicine treatment, whether it is all the plants are converted to haploid, or there are some mixture and still?
Actually, there is a process. Once the plant is haploid, it well establishes and it produces the tillers. So you are not giving the colchicine treatment to the seed, developing embryo. You are giving colchicine treatment to the root of that. You will cut the haploid plant into off. You will remove everything which is already reproductive or something. You will cut it. So it was there in the photo. You will cut it and dip that in the colchicine solution. We normally use 0.01 to 0.03% of colchicine. When you dip that into the colchicine for 3 hours, so what happens is, plant has been cut already, and the root will start absorbing the colchicine into its system. So instead of converting or giving colchicine to the reproductive tissues, we are directly making the plant to absorb it and taking it into its blood. So every tiller, every root, every leaf, every cell of that plant will be converted. And especially, even the established haploid plant may not be doubled. Whatever regenerates from there, cell production happens, then tiller comes, then reproductive organs will be formed, seed will be formed. For all that process, you have to have a mitosis and meiosis. So that in all those new cells created, the chromosomes will be doubled there. So whatever the seed you get, that will be having a doubled chromosome numbers. Yeah, that's. So you're creating stress for the plant to absorb colchicine into its blood. It's not giving specific injection like what we give it for making stun, or some, if you want to remove teeth, they give to some particular area. Just this will be made it very not sensible. But you're giving it the whole body when you do operations. That's the difference we have. So whole plant system is treated with that colchicine. Anything else? Are we done? नाइस है, ठीक है, थैंक यू सो मच, थैंक्स फॉर कॉलिंग। थैंक यू। Thank you so much. Thanks. I got a pen drive. Yeah. हाँ वो एक्चुअली ना थोड़ा फर्स्ट हाँ वो क्या हुआ था, वो एक्चुअली 1 महीने से पहले मुझे तो मेल आया था।
नहीं नहीं, वो मेल नहीं आया था, वो